Hi, I'm a postdoc in Junji Go's lab at Yale University. The study I'm going to introduce today is about identifying a small molecule targeting SARS-CoV-2 protein translation. The SARS-CoV-2 genome is a 30 kb single-stranded RNA. Following entry into host cells, the virus releases its genomic RNA and immediately starts translation of a large open reading frame, or F1AB, which encodes the non-structural proteins critical for viral transcription and replication. RF1 consists of two parts, 1A and 1B. And RF1B is actually in the -1 reading frame of RF1A. So the way that RF1B being translated is that the ribosomes have to undergo a -1 frame shifting at this short overlap of 1A and 1B. Before go on translating RF1B. This -1 frame shifting is mediated by a frame shifting stimulating element, or FSE. The FSE contains a 7 nucleotide slipper sequence and the 3 stem pseudoknot structure. During translation, the pseudoknot structure temporarily stops translating ribosome and creates tension between the viral RNA and aligned tRNA. Then a subset of ribosomes would slide 1 nucleotide backward at this slippery sequence when tRNA realigns in the -1 reading frame. Any non-frame shifted ribosome will terminate at this stop codon in frame with RF1A. By comparing FSE between SARS-CoV-2 and SARS-CoV, we found that they are highly conserved and only differed by this single unpaired nucleotide. Besides SARS-CoV and CoV-2, frame shifting is widely adopted by all coronaviruses and many other RNA viruses in their protein translation process. Considering the high conservation of frame shifting events across many viruses, we reasoned that perturbation of frame shifting would be deleterious to these viruses, and finding small molecules targeting frame shifting could benefit both studies on the mechanism of frame shifting and the development of therapies. To make a frame shifting reporter suited for high throughput compound screening, we put the SARS-CoV-2 frame shifting stimulating element between the M-cherry and the GFP coding sequences, with GFP in the -1 reading frame. So the ratio between GFP and M-cherry signal would report the frame shifting frequency. Then we transfected this frame shifting reporter into HEC293 T cells. So compared with the no frame shift GFP fusion control, the frame shifting reporter showed a reduced but still robust GFP signal. After we move the GFP into the 0 reading frame, we lose all GFP signals, which is consistent with the notion that any non-frame shifted ribosome will terminate as the in-frame stop codon. After teamed up with Yale Center for Molecular Discovery, we screened more than 4,000 compounds, including both FDA-approved drugs and the drug candidates currently at clinical stage. So we treated HEC293 T cells with each of the compound and then transfect the frame shifting reporter into the cells. And after 24 hours, we took image and calculated the GFP to M-cherry ratio. From the effect size distribution, we found that the vast majority of the compound had very little or no effect on frame shifting. Most of the screen actives were false positives due to their intrinsic autofluorescence. And they were ruled out by manual inspection of image. And from the compound screening, we identified a robust frame shifting inhibitor, merofloxacin. It belongs to a big family of antibiotics known as fluoroquinolones. They inhibit bacterial growth by targeting their DNA topoisomerase and DNA gyrase, which are needed for genomic DNA replication. But interestingly, none of the other fluoroquinolones that we have tested showed inhibitory effect on frame shifting. So we next tested the dose-dependent effect of marofloxacin on SARS-CoV-2 frame shifting by using the luciferase-based frame shifting reporter assay. And we found a progressive inhibition of -1 frame shifting as we treated cells with increasing concentrations of marofloxacin. And 50% inhibition was observed at about 20 macromolar. To test whether the effect of marofloxacin may be weakened by mutations within the frame shifting stimulating element, we introduced sets of synonymous mutations intended to disrupt the signal structure. Despite the wide range of effect of these mutations, the -1 frame shifting effect of marofloxacin was largely unchanged, suggesting that -1 frame shifting inhibition of marofloxacin is robust to moderate perturbations to the sequence or structure of frame shifting stimulating element. To further study the targeting specificity of marofloxacin, we continue to test a panel of -1 frame shifting reporter with FSEs from alpha, beta, and gamma coronaviruses, and from some other viruses, and also from the human pectin mRNA. And marofloxacin showed strong selectivity toward FSEs of beta coronaviruses over those of alpha and gamma coronaviruses. And the marofloxacin did not inhibit -1 frame shifting of the other viruses that have been tested, nor the human pectin mRNA. Now we have observed anti-frame shifting activity of marofloxacin in reporter assays. We next asked whether it could impede -1 frame shifting and viral growth in SARS-CoV-2 infected cells. So after infecting viral E6 cells with SARS-CoV-2, we treated cells with varying concentrations of marofloxacin and quantified the abundance of NSP8 and NSP12, which are encoded by ORF1A and ORF1B respectively. As expected, the expression of NSP12 was substantially reduced by marofloxacin. At last, we did plaque assay to measure the viral yield under marofloxacin treatment. We found that marofloxacin effectively impeded SARS-CoV-2 replication without causing substantial cytotoxicity. And when we plotted the relative viral titer against the relative frame shifting efficiency, we found a near exponential decrease in viral yield as the -1 frame shifting was increasingly inhibited. This result suggests that -1 frame shifting efficiency is rate limiting for SARS-CoV-2 replication, and targeting frame shifting could be an effective antiviral strategy. The molecular mechanism by which marofloxacin inhibits frame shifting is currently unknown. To study whether marofloxacin might directly bind the FSE of SARS-CoV-2, we and the other lab performed multiple assays, including circular dichroism, shape probing on SARS-CoV-2 FSE with the presence of marofloxacin, and also optical tweezers. And all of the assays gave us negative results, strongly suggesting that marofloxacin does not directly bind SARS-CoV-2 FSE. In a preliminary experiment, we tested marofloxacin in 2 different in vitro translation systems, the rabbit radiculocyte lysate and the wheat germ extract. While we found a strong inhibition caused by marofloxacin in rabbit radiculocyte lysate, the inhibitory effect of marofloxacin on SARS-CoV-2 frame shifting element was gone in wheat germ extract. We reasoned that the inhibitory effect of merofloxacin should due to interaction with endogenous factors, which are potentially different in mammalian and plant cells. Since merofloxacin's effect showed specificity toward beta coronaviruses and mammalian cells, the first suspect kept to our mind is the interface between viral FSE and mammalian ribosome. And we are currently doing experiments to look into more details. So in summary, merofloxacin inhibits mandatory frame shifting of SARS-CoV-2 and other coronaviruses. And the frame shifting inhibition by merofloxacin is robust to mutations within FSE. And merofloxacin impedes SARS-CoV-2 replication in infected cells. And going forward, I will perform ribosome profiling to understand how merofloxacin affects the translation of viral RNA. And I will also use chemical probing and shape map to check the potential interaction between merofloxacin and human ribosome. First, I'd like to thank my advisor, Junji, and all the lab members in the Go Lab for their help and valuable suggestions. Most importantly, I want to thank all my collaborators across EO. So Laura and Yulia, they performed the high throughput compound screening. And Wendy Gilbert's lab is currently helping me with ribosome profiling. And the people from Brett Lindenbach's lab, Yaqin Ho's lab, and Greg Whelan's lab, they all helped me with the measurement of viral yield. And also there is an ongoing project in the lab studying mandatory frame shifting regulation by a cellular factor. The project is led by my lab mate Lianhuan Wei, and she will have a poster during the meeting. So go and visit the poster like if you are interested. And thank you everybody for listening.